Welcome to Indoor Ag Science Cafe. I'm Chieri Kubota, The Ohio State University. So today we are, we are going to do the more basic stuff. Well, for those who are new to the cafe, welcome. This is a forum, open forum for the discussion space or chatting space regarding science and technology going into indoor farms. This whole activities are supported by USDA grant, NIFA grant, SCRI. So I'm using a new logo of that project. The project is led by Michigan State University with collaboration among many scientists in Ohio State, Purdue, and University of Arizona. So that's us. So again, the mute button is really important. So I just wanted to re-emphasize, you have a control. And then you can unmute and mute. If you're calling in, you may need to ask for permission. It's case by case. I don't really understand that. If that's the case, just text me so that I can unmute. So we are doing a new series starting December. And some of you, or many of you, already got this notification of or request or call for questions we can use for query series. So the query is basically expert, in the same space, and then answering to typical questions or good questions we gather. So I created the question site. So you submit any questions, either simple question or more complicated question. You don't need to, disclose your name or, contact email. You just type in the questions. You may be selected, you may not be selected, but that's the sort of regular opportunities to talk about questions of many different things: technology, science, economy, anything. So that's what we want to do. I have only a few questions so far, so I really encourage you guys to, put the questions to the query site. Right after this cafe, I'm going to send in another request, basically encouraging you to type in the questions. All right. So again, this is a conversation space and then also learning space. So today I'm going to give a more basic side of information. November Cafe. December Cafe is going to be the queries, right? And then January, we have 80-acre farms. Vice President Eric Ellerstedt, he's going to talk about the company, introducing what's going on in 80 acres farms. This is an indoor farm located in Cincinnati area. So something you might want to look forward to that. And then the February Cafe, we will have Dr. Paul Fisher from University of Florida. He's going to talk about biofilm development in the nutrient recirculation system. So that's something quite important whenever you are doing leafy green production or any crop production with recirculation nutrient hydroponics. March and April, I have openings, so anyone who want to volunteer is welcome, or request, potential speakers. I really appreciate. Again, this is a forum for you, so you got to say something, whatever you want to hear. All right. So, any questions so far with the plan, and then also maybe queries? I can take that now. All right. So as standard, we are recording the first part with presentation, and then we stop the recording so that you feel more comfortable to ask questions. But I like to encourage you to interrupt my presentation because I'm talking today, and then ask questions if you wish. But it's going to be recorded. I can't edit that. I don't have time to edit all these questions during the recording. But if you feel comfortable to be recorded, why don't you just ask questions in the middle of presentation? If you don't feel like comfortable, then if you're not comfortable, then you wait and save the questions after the presentation so that your questions and communication, voices, maybe faces are not going to record it. Okay, so today's topic, back to basic, plant response to temperature. So I wish I have more specific research data for this, but the original purpose is just to give a basic understanding so that everyone is in the same foundational understanding about temperature response of the plants. So I thought this is helpful, particularly those who are not really trained in this controlled environment plant physiology area. Again, this is a new logo of the project. Okay, so the temperature is very important. Everyone knows about that. We provide optimum conditions for plants. Typically, root zone and then aerial zone. Temperature is one of the most important environmental factors. So that's why we pay attention to temperature in addition to other factors. So today you're gonna go over some of the basics. Number one, plant temperature is different from air temperature. It's something very important to understand. And number two, understanding daily average temperatures, sometimes overlooked, but it is important to pay attention to that daily average or the 24-hour average or average over a certain period of time while controlling daytime and nighttime temperatures. Everyone pay attention to daytime and nighttime temperatures because that would affect directly photosynthesis and respiration and other things. But sometimes we'll get daily average temperatures. So I want to emphasize that. And then also, you probably know cumulative temperature. Sorry, go back again. You probably know optimum values are species and cultivar specific. That makes the more challenging whenever leafy green growers need to work or produce multiple crops, multiple species, multiple cultivars together. But that's important to understand. And then research hasn't caught up the needed information regarding species-specific or cultivar-specific response. We certainly need to contribute to that. The third point you're going to go over quickly is the cumulative temperatures to understand cumulative response. And then sometimes over days, you can integrate to understand the cumulative response. And then bottom line is more research is necessary, and that's why we are working. All right. Oh, by the way, the background figure, you might wonder what it is. It's actually speaking letters. I copied from kids' YouTube video site because I'm going to talk about speaking letters concept toward the end. So that's why. So this one is quite nice chart. Contribution from Dr. Toshio Shibuya from Osaka Prefectural University. Many years back, he did this research under the electric lighting, artificial environment conditions in growth room, and then looked at leaf temperature difference at varieties of conditions. This particular one is the change of leaf temperature relative to air temperature, but air temperature was very constant temperature. So basically looking at leaf temperature increase during the time plants need more irrigation. You don't see any wilting or anything, but if you measure the leaf temperature accurately, like he did, you can detect as great as 9 degrees Fahrenheit the increase during the time plants sort of reducing transpiration. So I thought this is a great presentation showing that air temperature and leaf temperature are not always the same, or most of the cases they are different. All right. So understanding air temperature versus plant temperature. As I said, plant temperature is not the same as air temperature. And then even under the constant air temperature conditions, and then radiation may be the constant, like indoor farm does provide. And leaf temperature varies at different level depending on many factors. And then I come back to what kind of factors involved to affect leaf temperature in the next slide. So theoretically, leaf temperature is more important than air temperature, particularly if you're looking at photosynthesis or transpiration, something happening in the leaf. If you are looking at development side, meristem or growing point temperature is more important. Flower initiation, things like that is coming directly from meristem temperature. So that's a theoretically, right? And then researchers usually pursue to acquire leaf temperature information, sometimes even meristem temperature information using a teeny tiny thermocouple sensor going into the growing point. But realistically, accurately measuring plant temperature, leaf or meristem is very difficult. Leaf temperature you can measure with thermal camera, but there's an emissivity issue. Emissivity is something affecting temperature reading. So unless you really unless you just want to know the relative difference, between surrounding place and plant growing area, unless you have good, capacity to calibrate, often the resulting measurements are not accurate and then not so useful in a scientific application particularly. And then also another thing is complicated is even within the same plant, same leaf, for example, temperature are different. Temperatures are different depending on the location, depending on the size of the leaf. For example, upper leaves versus lower leaves, leaf closer to the air circulation area, or it's depending on the surrounding microclimates. Even within the same plant, you may see the different temperature. So that chart on your right, this one is really nice chart. Again, captured by thermal camera, infrared camera, shared by Dr. Kitaya, also from Osaka Prefectural University. And he's showing, I think it's a sweet potato leaf sitting in an environment, a quite constant environment in terms of light and temperature, and laminar flow going from upper left corner to lower left over the surface of the leaf. And then sweet potato leaf is very flat, so that's why he's using that as a model system, but showing nicely where the within the leaf, first of all, a huge difference in terms of temperature. So it's showing like 28, 27 degrees C, all the way to 32 or 33 degrees C within the same leaf. So the 5 degrees C difference, that means 9 degrees Fahrenheit difference again within the same single leaf. So the conditions affecting that is radiation, air temperature, and air current speed, and then also leaf size. And then this actually, because of the leaf size, relative to farther away from the inlet of the air, is affecting the increase of temperature. And then, of course, factors affecting stomatal opening, because transpiration is the mechanism to cool the temperature down. So anything limiting transpiration, anything limiting stomatal opening would reduce that cooling effect and then increase the temperature quite a bit. So those are the things you need to understand. So that's the importance of leaf temperature or plant temperature and air temperature. Now, so if you grow plants, you want to start with selecting a temperature for daytime, a temperature nighttime in your indoor farm. And then you hope to have a cultivar-specific recommendation, which usually doesn't exist. We have genetic information like lettuce, tomato, basil, some crops, what's the day temperature, what's the night temperature. And then I gathered some of the information out there and then also anecdotal information. One good example showing here, the importance of temperature done by Dr. Chris Curry at Iowa State University with his former graduate student, Kelly Walters, showing that really nice response of temperature of basil. Basil tend to like warmer temperature, and then that's kind of genetic information we have now. And he was demonstrating that very vigorous variety, Nufa, basil, having a optimum temperature around 29, 30 degrees. We wish to have many information or many different varieties-based information like this uh, uh, to help indoor growers to select, but the reality is we don't. And then new cultivars coming in. So basically using your facility or using collaboration with like a university having many different growth chambers, you really need to get that info. So let's talk about basics because we don't have specifics much. So the temperature, again, it's very important. It's a driver for all biological processes. Um, there are 4 typical variables we want to use to evaluate the temperature environment. So number 1, daytime temperature. As I said, photosynthesis and transpiration rate are affected by daytime temperature. Nighttime temperature affect dark respiration, so the consumption of carbohydrate at night. And then something very important is average daily temperature. That's a 24-hour average typically. And then that is determining development rate, plant development rate, such as rate of increase of number of leaves or days to see initial flowers. So those are the development rate related phenotypes. And then that is pretty much affected or pretty much explained by daily average temperature. So right hand side is showing a typical temperature response of the development rate. Growth or photosynthesis look similar to that, but actual temperature range may be slightly different. But anyway, so the important thing you need to understand in this graph is there is a minimum temperature, and below that you don't have any positive growth or development happening. And then from that point, usually almost linear increase as increasing temperature. So developmental response reach maximum at the optimum temperature. So the mountain shape is depending on, I guess, the observation values, whether it is number of leaves or photosynthesis or species dependent. It may be more sharp peak or it may be more flat peak. But anyway, so there's an optimum temperature and minimum temperature, and then between those two temperatures, typically called linear range of temperature response, which is very important to understand in the further understanding plant response. Also, we often talk about day-night temperature difference. I'm gonna talk about that in a minute, so I'm gonna skip this. But it's basically affecting morphology of the plants. All right, so I wish I have something like this for the horticultural crop, but this is a agronomy crop, so the cottons and peanut and rice and things like that. But it's a great way to show, the different optimum temperature. So every single one is a crop specific genetic temperature response curve showing optimum temperatures somewhere between 20 degrees C, which is like a 68 degrees Fahrenheit, all the way to 33 degrees C, right? So that's 90 some degrees Fahrenheit. And this chart is sort of normalized based on the temperature 20. So everything at 20 is showing the rate of 1.0, so the relative value. And then the rest of the response is showing relative to that magnitude of response. So that's why if the optimum temperature is really close to the 20, then the mountain shape is shallower. If the optimum temperature is really high, like a 30 some degrees, then you have a huge, response. But again, the important thing is the optimum temperature and shape of the plant response to the temperature is pretty much crop specific and then even cultivar specific. All right, so some of the example of how to think about temperatures in this slide. So again, day temperature affect the photosynthetic rate. So right hand side is a chart I extracted from one of the physiological works previously done by someone. So it's a textbook type of information, how respiration, gross photosynthesis respond, therefore net photosynthesis respond to the temperature. So just the temperature is relatively odd range because this particular one, photosynthetic optimum is quite low, 15 degrees C or so. Usually leafy greens are having 20, 25, maybe a little bit even higher range. But don't worry about actual temperature, but shape of the response. So as you know, plants do photosynthesis, which is called gross photosynthesis, true photosynthesis, and then doing respiration at the same time, dark respiration, photorespiration. And then respiration typically respond like an exponential way with increasing temperature. And then the difference between gross and respiration is basically this net photosynthesis. That's the contribution of the carbohydrate production, contribution to the carbohydrate production. That means biomass accumulation of the plants. That means yield of the plants. So anyway, so the selecting optimum temperature daytime, looking at this optimum range is always important. And then the quite, similar to the previous temperature response curve, there is a linear range of the temperature. And then, for example, if you see the optimum temperature 15 degrees C, and then if you start thinking of reducing temperature 10 degrees C, nobody want to do that, but let's see if you increase 10 degrees C, the photosynthetic rate reduce 50%. So that is really, the temperature sensitive. The mountain shape may be a little bit more flat in the actual case, but in that case, you can accept a certain range of temperature without worrying about a sharp reduction of photosynthesis. And then again, this needs to be collected, understood based on your conditions and then also species cultivars. So one thing I get often asked is nighttime temperature selection. Whether it is a good thing to reduce nighttime temperature so that, for example, carbohydrate waste or consumption is reduced, therefore yield might increase. So if you look at this as a model response, right, photosynthesis and response, respiration, photosynthesis is much higher level than respiration. And then if you reduce the temperature and then reduce the nighttime respiration, for example, you can reduce quite a bit the same way, half of the respiration in that situation. But because of the level of respiration is already 20% or so of the net photosynthesis, so that during the night respiration is going on, but the magnitude of the respiration during the night is, in this case, about 20%. So 20% to 10% by reducing 10 degrees C, it's still, you might consider significant, but it's not like photosynthesis reduction or increase by 10 degrees C. Temperature difference. And then also most of the facilities, you're growing plants in very long photo period, like 18 hours of photo period. So the contribution of the nighttime is very small. And then if you do the math based on this response curve, reducing respiration from the same as daytime temperature, so that set the nighttime daytime temperatures the same, so 15 degrees C in this case, reducing that to 5 degrees C, for example, is only generating 2 to 3% of potential increase of daily assimilation, because nighttime is only 6 hours, and then 18 hours is going photosynthesis. So that's why if you test the lowering nighttime and then not seeing much improvement of the yield, maybe because of that. But if you care about 1% reduction or 1% increase of yield, then that's something you might consider worth trying. Something you can't forget, as I repeatedly said, you need to pay attention. You need to pay attention to the average daily temperature, because lowering nighttime, even 6 hours, lowering nighttime temperature, even short period of time, would definitely affect the average daily temperature or 24-hour average. And so you really want to maintain day temperature to cancel out nighttime temperature reduction, or make sure that average day temperature doesn't change. So that's something I really want to add if you didn't understand this importance of average daily temperature. Okay, so you might wonder, greenhouse, usually nighttime is almost always lower than day temperature, and why? And so there are several practical reasons. One, because energy loss during the nighttime is huge, so growers want to reduce the temperature as far as not affecting plant growth. So that's one thing. And then also daytime temperature typically gets higher, particularly warm season. It's very difficult to get down. So the lowering nighttime temperature is easier. So that's a result. Usually you have higher day temperature than night temperature situation. But when you pay attention to average day temperature and then keep that optimum, and then reduction of night temperature can generate increase of biomass, and therefore it's actually beneficial for those who pay attention to 1% potential increase, like tomato growers. 1% increase by whatever is very important for them. And then also lower night temperature can increase quality of flowers and fruits, flavor, size of fruit, size of flower. So that's why important to maintain the nighttime temperature relatively low. But for indoor leafy greens, I'm not sure if lower night temperature may not may be important or may not even necessary. So I'm not sure about that, but we need more research on this. It might be quality related factor also. We can discuss about that later. All right, so what about diff? So I mentioned about diff briefly. Diff is interesting technique to control the morphology. A lot of studied. Well, actually, the diff was invented by the work done in the Royal Hines lab at Michigan State University, and then used for pretty much the height control of the ornamental floriculture crops. And this is a study example, different diff and extending, at higher diff. And then that was applied in many different species. So they did find many species respond to diff. It's quite interesting, day and night temperature control plant shape. So there's an argument, or there's a question, diff might affect leaf expansion of some species because it's a sort of extending growth, the same as stem. But we know more research is needed because I couldn't find conclusive understanding of diff impact on leaf expansion. Some say yes, some say no. So that's something we want to know by doing a lot more research. One thing we know is the daily average temperature, ADT, affect leaf expansion. So if the ADT is high, the leaf expand quite fast. So this is a average daily temperature. This is the leaf expansion rate, showing that quite a bit of increase toward the optimum temperature. And this is not the leafy green data. This is a leafy green data. Showing 21, 24, 27, 30, 33 degrees C. This is actually light interception. So interception meaning more leaf in a space. So you can see that it's a little bit vague or unclear copy, but 27 degrees C, it's the fastest, followed by 30 degrees C, 24, 21, 33. So its response is almost like, increasing temperature all the way to 27, and beyond that, it's declining in terms of leaf expansion rate. So that's quite interesting. You can probably use this understanding when you need to accelerate leaf expansion when plants are maybe young, and then change temperature within the optimum range so that you can achieve something else other than leaf expansion as soon as maybe canopy close or something like that. So temperature is quite interesting and then useful to control. All right. One more thing I would like to inject here, or a couple more things. I hope to finish in a minute or two, but so plants respond in an integrating manner. So as long as in this linear range, average daily temperature is in the linear range, fluctuation do not really affect plant growth and development. Even instantaneous temperatures, if it is in a linear range, for example, photosynthetic response, right? So that's an instantaneous temperature base. If it is linear range, and then going up and down doesn't matter as long as average temperature is in the optimum. So this is actually the base of heat unit model. I explain the next slide, but right-hand side is, so again, this is not the leafy green, but showing the, the plant response in terms of days to flower. So the development rate is explained with one single unified model, regardless of actual daytime and nighttime temperature, because those temperatures are both in a linear range. Therefore, all the plant response of the development is explained by average daily temperature. So that's the cumulative way, the concept. So, in agronomy and crop science use this measurement quite a bit. Heat unit, degree days is widely used to predict the phenological stage, like a flowering stage or harvestable stage. And this can be used more effectively in an indoor climate because there is no disturbance like light or humidity, because you can control those things theoretically really nice. So, but anyway, if you're not familiar with heat unit, we always work in the linear range because if it is linear range, then temperature up and down is pretty much canceled out, and then everything can be, the cumulative response like growth and development can be explained with cumulative temperatures. But we have to consider the minimum temperature. So the average daily temperature minus minimum temperature is the daily heat unit, and then accumulating that every day is the cumulative heat units, and that can represent the potential growth to the certain stage. How you use this in the next slide. So for example, you have successful production at 20 degrees C, which is 68 degrees Fahrenheit, 42 days to harvest this whatever the crop, maybe leafy lettuce. And then you know that lettuce has base temperature about 5 degrees C. So use that information. You know the cumulative heat unit for 42 days to harvest is 630 degree days. So how did I get 630 degree days, which is basically 20 degrees C, average daily temperature minus base temperature 5. So that yields 15 degrees C multiplied by 42 days. That's 630 degree days. If you have to reduce the temperature to 18 degrees C, maybe because you have Mizuna growing, or maybe because you have something kale or some brassica that requires a lot lower temperature. Maybe strawberry growing. Strawberry's optimum is 18 degrees C. And then you want to see the impact on your leafy greens, lettuce. And then in that case, you can use this relationship, 630 divided by the updated new temperature, 18 degrees C minus base temperature. That gives approximate duration to reach the same developmental stage, in this case, harvestable stage. And then in this case, the value is 48 days. So that means you have to experience about 6 days of difference delay. So it's quite interesting, you know, you can use this effectively. And then also this demonstration showing that 2 degrees C difference, you might think it's a minor difference, but actually yielding quite a bit of delay in terms of harvest. So it's quite important. So this concept of integration is quite interesting. As long as average over integration time meets the target average temperature, for example, you can extend that period. There are 2 quite interesting research reported years ago. Tomato integration temperature can be not daily, but expanded to 12 days integration time. Chrysanthemum was demonstrated with 6 days of integration time. And then that, actual fluctuation within that period didn't affect the growth or morphology or any important parameters of those crops. And then I want to do that in the lettuce also, because that gives flexibility to control and opportunities to do quite interesting way to control the temperature and then also production. If you are affected by outside conditions, maybe like greenhouse, then you can actually do opportunistic control or anticipatory control. So the predicting the next few days of potential temperatures and then include that in a decision of today. Another one is quite interesting, inspired by working experience with this I would call genius engineer, Takehiko Hoshi, who is not known so much in this space, but he did a quite interesting way to do plant production. Basically, plants cannot move and staying in the same place. Therefore, you have to control temperature, you have to create the uniformity. But his concept was, if plants can move or plants can be in a system that is movable, why don't just listen to the plants and then move the location from something not so optimum to something more optimum. So that's sort of the concept he demonstrated in a project I worked with at Chiba University, gosh, 20 years ago. So that's I think applicable in the indoor farming also. All right, so I think that's the last slide. Yep, and then I really appreciate listening to me. And this is my lab members, and then we are excited to do more work in the indoor. Many of the Optimia project team funded by USDA will be working on optimizing controlled environment, and temperature is going to be there one of the factors. So we are looking forward to gain or acquire more knowledge from their outcomes. So let me stop recording.